also practice other questions related to the Hardy-Weinberg law. Okay. Multiple alleles. Slide change भएको छ? भछ?
छैन सर।
छैन? ओके, अहिले त भयो होला? ओके, अब हिजो हामीले सिंगल जीन लोकस विथ 2 एलिल को बारेमा कुरा गरेका थियौं. How these alleles are being present in normal autosomal or sex chromosomes, and what type of phenotype will be produced through the crossing, and is there alleles they are present in the equilibriums after generation to generations with if there is any evolutionary process are acting. त्यसको बारेमा हामीले हेरेका थियौं हिजो. र if there is a random mating being happens, or there is the panmictic populations, or there is the random mating population is being exist generation after generation in the original population, if there is the Hardy-Weinberg law exists, or generation after nth generations, still there are individuals with of different kinds of the genotypes are changed, but the frequency is remains constant. Whether it will be remains constant or not can be tested by utilizing the chi-square test. Whether the population is present in equilibrium or not will be tested using chi-squares. त्यसको बारेमा हेर्दै आज र यो मल्टिपल एलिल्स. अब 2 एलिलिक सिस्टम diallelic system ma certain genes they are found to be governed by the multiple alleles that we have been found, just like is the ABO blood group, MN blood groups, which are found to be of multiple alleles. If there are multiple alleles being present in a single locus with the ABC, RPQR, STU, BW, like this alleles, if it is a pandemic populations, are these multiple allele are also exist in the Hardy-Weinberg law or not, and how can we calculate their gene frequency or allele frequency if there is the multiple allelisms is governed by this multiple alleles topics. So when more than 2 alleles are present at a locus, the equilibrium condition is described by the multinomial expansions. इस हामीले बाइनोमियल एक्सपेन्सन को p + q को स्क्वायर को बारेमा बाइनोमियल एक्सपेन्सन को भन्यो भने मल्टिपल लोकसलाई चाहिँ मल्टिनोमियल एक्सपेन्सन्स चाहिँ whether it is present in the equilibrium condition or not भनेर चाहिँ multinomial expansion will describe about this. So just like as the p + q + r + this को square in which each letter represent the gene frequency of one allele. First allele को gene frequency p, second allele को q, third allele को r, fourth को s, fifth को t. In this way there will be the frequency being represented. For example, for 3 allele conditions with A1, A2 and A3 with the respective frequency p, q, r, we would expect 6 genotype at Hardy-Weinberg equilibriums. When there are 2 alleles, there are the 3 genotypes. When there are the 3 alleles, there are the 6 genotypes being expected at the Hardy-Weinberg law in the proportion as p square A1 A1, 2pq A1 A2, 2pr A1 A3, q square A2 A2, 2qr A2 A3, and r square A3 A3. If all the alleles are codominant with each other, each genotype has its own distinct phenotype, and the genotype frequency are easily scores. 6 different genotypes exist in the Arden-Ward law if there are the 3 alleles being present. If these alleles are co-dominant, every 6 genotype they have their unique phenotypes and with distinct kinds of the phenotypes expressed by this, and their genotypic frequency they can be easily computed from the populations. How this will be computed, that P square A1 A1 to PQ A1 A2 to PR A1 A3 जसरी हमले जो P or Q in one sides male sides, P and Q in the female sides calculate the relations between the genotype and gene frequency calculate. In the same way, you can expand this as male side में PQR, female side में PQR. By P for the A1, Q for the A2, R for the A3. The checkerboard development one, you can easily calculate these frequencies: P square to PQ to PR, Q square to QR, and R square for the 6 different genotypes. You can easily calculate the things. And from this genotypic frequency, you can calculate the gene frequencies. As P is equal to 2A1 A1, 2NA 2NAA, NA and plus NA heterozygous NANA locus upon 2N using formula ने हो हामीले अगाडि हेरे चाहिँ दुईटा भयो भने के हुन्छ 2NAA र heterozygous NAA upon 2N हुन्छ भने अब यो ट्रिपल भएको हुनाले मल्टिपल एलिल with 3 एलिल भएको हुनाले extra 1 is being genotype being comes here. So P is equal to A1 A1 plus A1 A2 plus A1 A3 upon 2N. Q is equal to 2A2 A2 plus A1 A2 plus A2 A3 upon 2N. R is equal to A3 A3 plus A1 A3 plus A2 A3 upon 2N. We can calculate the gene frequency. Where A1 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 A2 A1 A3 etc. refers to the number of genotypes in each category. N refers to the total number of individuals scored. So in this way, multiple alleles, if the gene is governed by the multiple alleles, we can be computed how the gene frequency are being present in that population and what kinds of the genotype will be produced by the mating system. So in this way, we can easily calculate the things. Okay, then another is the factor affecting the gene frequency. Arden-Ward law exists when there is no evolutionary forces acting upon the populations. If the evolutionary forces are acted, then the frequency are changed. So how this frequency are being changes due to the different evolutionary forces. One is the mutations. We are well known about that. The mutation is one way for development of the variation in the populations, and the mutations maybe happens in the forward direction as well as reverse direction. Particular genes then can be mutated. Okay, so there is no dependency over the other genes for a mutation to be happen for a particular genes, and point mutation or any kinds of the mutation maybe takes place there. So the gene frequency is affected by the frequency of mutations that introduce a particular gene into a population. So particular genes may be introduced in the populations so that the frequency of the genes will be changed in the populations than the original population. Our original population, if we consider a single locus with 2 alleles A and A, and we consider dominant A and recessive A, if we considered, then the dominant A can be continually changed to the recessive A, and the recessive A can revert back to the dominant A. Both forward and reverse direction maybe mutation may be happen there. So generation to the generations after the frequency of this allele will be changed if mutation is being happens. It is in dominant but recessive allele mutation by forward mutation by next generation the frequency of the recessive allele will be increased and the frequency of the dominant allele will be decreased. That will be happens. So if the mutation be happens for the nth generations, if we consider n generations, a single generation by one the original frequency for the dominant A डोमिनेन्ट ए को लागि ओरिजिनल फ्रिक्वेन्सी p0 छ र रिसेसिभ को लागि चाहिँ q0 छ भने हैन र रेट अफ फ्रिक्वेन्सी दिस रेट अफ म्युटेसन्स अफ दिस डोमिनेन्ट allele to convert to the recessive allele if considered be u. It is the rate of mutations, that is dominant allele to convert to the recessive alleles. Then after the n generations, the frequency of dominant A becomes original frequency p0 upon 1 minus mutation right to the power n. This becomes the frequency of the allele A. Okay. So it will be changes 1 minus u to the power n times the frequency of dominant allele will be reduced and will be reduced in the n generation of the population. And even if the mutation rate of A to A is small, can get dominant A to the recessive A mutation, if it is very small odds, then after a large number of the generations, and n tends to the infinity generations, the A is equal to p0 1 minus u to the power n becomes 0. This value becomes 0 after the n generations, and 0 becomes 1 minus 1. There will be the value that 1 minus 1. That means 1 minus 1 0 0 to the power n 0 0 into p0 0. So A becomes 0. That will be happiness. So इसका मतलब क्या है? All the dominant A convert to the recessive A. At that times, the value becomes 0. When all dominant A convert to the recessive A, at that time the value of this becomes 0. So that after the n generations, the A will be disappeared. And n becomes infinity. So mutation rate does not always occur in only one directions. The ratio of forward and backward mutation may not be same. Again, अब फरवर्ड म्युटेसन मात्र हुँदैन पपुलेसनमा त ब्याकवर्ड म्युटेसन पनि हुन्छ. So that the dominant allele convert to the recessive allele as well as recessive allele convert to the dominant alleles, but the dominant to recessive forward and backward mutations, the rate is not similar. Another way, so the actual number of genes mutating depends upon the rate of mutations and the frequency with which the allele is encountered in the populations. Number of genes mutating में depend करता है rate mutation में, क्योंकि forward mutation को एक किसी को rate होता है, backward mutation को और किसी को rate होता है, both are not the similar. Same rate में होता है ना. So the the alleles frequency being predominant over the populations. A dominant allele high है कि recessive allele high है कि that. So that actually describe the number of genes to be mutated. And with every forward mutation of dominant to recessive, the frequency of decrease and that of A increases. As more A's are formed, the total number of back mutations will increase against. A dominant बाट recessive allele में convert हुँदै गयो, forward mutation हुँदै गयो. Just like forward mutation हुँदै जाँदाखेरि nth generation में के हुन्छ भन्दा recessive allele frequency बढ्छ. अनि अर्को साइडमा के हुन्छ भन्दा recessive allele फेरि backward to the dominant allele भएर आउँछ. जति recessive allele frequency बढ्दै जान्छ त्यति नै the backward mutation rate also increase in the same ways. Backward mutation फेरि increase भएर फेरि dominant alleleहरु फेरि formation भइरहेको हुन्छ. So that the dominant allele becomes completely disappearance is a very rare events. So one times अब हामीले के भन्छ भने त्यो चाहिँ backward र forward mutation चाहिँ equal हुँदैन, तर one times र forward बाट forward to backward mutation भइरहेको हुन्छ, dominant to recessive allele formation भइरहेको हुन्छ भने recessive allele to dominant allele पनि formation भइरहेको हुन्छ. अनि so one times there will be a point will be comes, equilibrium points will be comes. त्यो पोइन्टलाई हामी के भन्छौँ भन्दा म्युटेसनल इक्विलिब्रियम भन्छौँ. फरवर्ड टु ब्याकवर्ड म्युटेसनको एउटा पोइन्ट अफ इक्विलिब्रियम विल बी मेट इन one times in the populations in a temporal dimension. So eventually an equilibrium will be reached where the number of forward mutation is equal to the number of backward mutations. That point is called the mutational equilibrium. If the backward mutation rate is B. The backward mutation or the reverse mutation rate for recessive to the dominant converts is B. And the initial frequency of dominant is P naught. Initial frequency of recessive is Q naught. If this is the case, then single generation mutation will produce a frequency of dominant A equal to P naught plus B Q naught. P naught plus B Q naught. And the frequency of A becomes Q naught plus U P naught. That will be happens in single generations. This is the rate of backward mutations for the recessive alleles. This is the rate of forward mutation for the dominant alleles. So this frequency when added to the original frequency, it becomes the dominant allele frequency. This frequency added to the recessive frequency, that becomes a frequency for the recessive alleles. At the mutational equilibrium, which will be established, then when this mutational equilibrium will be established, when there is BQ minus UP is equal to 0 or BQ is equal to UP. The BQ is equal to UP भएको अथवा BQ minus UP is equal to 0 भएको conditionalमा mutational equilibrium हुन्छ. And since Q is equal to 1 minus P, then BQ भनेको 1 minus P is equal to UP. So B is equal to when you send it on other sides, what we get B is equal to P U plus B. B minus BP, you can open it, B minus BP is equal to UP, B is equal to U plus BP, and PP is common and U plus B. So B is equal to PU plus B, and then the P is equal to B upon U plus B. So the frequency of the dominant allele becomes B upon U plus B. In this way, we can calculate these things. So this shows the mutations. Because of the mutation being happens, the frequency of the allele are being changes at the nth generations, and it will be not remains same frequency whatever being found in the original populations. That's one way, one factor that will causes the changes in the frequency, even though there is a random mating populations is being analyzed. About factors that will affect is the selection practices. Selection ta both natural as well as artificial today huncha. Artificial selection one kinds of the changes later will be talk about this. Artificial selection, natural selection particularly exists in the natural populations that also causes the changes in the frequency of the allele being present in that population. So our selection is related to the reproductive abilities. So related to your selections. Individual selection in the nature means those which have reproduction ability is good or they are adaptive to the changes in environment, they can exist. And those which cannot adaptive to the environments, they will be exit from the populations, and that will be happening. That is related to the rate of reproduction. So that's the selection is defined as the differential rate of reproduction of different genotypes in a population which is measured as fitness. I'm going to fitness group measured. So it is the differential rate of reproduction of the different genotypes present in the populations. Different genotypes with different kinds of the reproduction rate also. Not all individuals have the same kinds of the cell reproductive efficiency, and they do not have the same kinds of the fitness. So they have a differential rate of fitness also. The fitness group measured rate of reproduction. So the change in frequency of the gene lies in the ability of its carrier to produce surviving offspring. Frequency of the gene could changes. What is the reproductive ability of that carriers which carried that genes? And what is the ability to reproduce the survival of offspring? If individuals carrying one alleles can produce the survivals offspring, then the allele frequency will be present there for the particular genes. If it cannot produce the viable offspring, then the frequency of that allele will be reduced. or it will change this. So in crop improvement, selection is very important because it allows the selected genotype to reproduce, while the undesirable genotype eliminated. The breeder is able to improve the various character. And generally plant breeding selection practices or artificial selection practice. Selection garda favorable allele selection garera. So unfavorable allele discard garera. So next generation the allele of the favorable genes will be increased. The frequency of the favorable allele being increased, while the unfavorable alleles they becomes decreased. So that is being practices here. Only those individuals which are being selected, they will put in the mating system, and those discarded will lost from the population. Our selection. So to be understand these things, first you must have to consider a single gene locus that having two alleles, dominant A and recessive A. If there are the two alleles for a single gene locus, it will produce the three kinds of the genotype: dominant, homozygous, heterozygous, and homozygous recessive. So selection may be practiced towards the dominant homozygous, may be selected. Selection may be happen to the heterozygous dominant, or selection may be happen to the homozygous recessive sites. And whenever the selection being practices, that will cause the changes over the frequency of the particular alleles differently. So just like this, selection may regard as scrutinizing process that rewards vigorous varieties of crops with reproductive successes and helps explain how new varieties species survive and increase. Our selection just like the scrutiny is there. जो बेस्ट हुन्छ त्यसलाई हामी सिलेक्सन गर्ने भन्छौँ लाइक दिस हैन अब अर्गन के अंदर अब हाम्रो इन्ट्रान्स सिस्टम भने जस्तो दोज व्हिच क्यान कम्पिट अर दैट क्यान ब्रेक डाउन द कट अफ पॉइंट and that comes in the forward, they get the admission. Those which are being backward, they cannot get the admission. So scrutinize here. Selection ma pani hami breeding practice ma hami selection gareko, we will scrutinize the genotypes that contain the different kinds of the alleles. Those which are being selected, they will be put in the reproductive mating system, so they become success. And also it helps in the explain the new varieties and the species, how they survive and increase in the population that can be explained by the selection. So selection works on phenotypes, not only individual genes. Recombinations due to sexual reproduction will also provide materials for selection process. Our selection I mean different type based selection also not based on the genotypes. Genotype based हामी selection गरेर phenotype based हामी selection गर्छौं र individual gene को आधारमा हामी selection गर्दैनौं कहिले पनि breeding मा particular genes लाई हामी selection गर्ने भने हामी selection गर्दैनौं phenotypic trait evaluation को आधारमा हामी selection गर्ने गर्छौं अनि त्यसैले चाहिँ जुन हाम्रो recombination हुन्छ अथवा क्रसिङ ओभरले गर्दा जो recombinantहरु डेभलप हुन्छ that also provide the opportunity for the selection to be happening in the population. So mechanism of selection are varied, such as temperature and humidity fluctuation, changes in the ability of the food, differences in the sexual attraction, and many more subtle influences. So selections mechanism variety about the different factors affect on the selection mechanisms. Temperature changes, reproductive relative humidity changes, so that is of climatic factors. The food availability, differences in sexual attraction, and there's like is of the factors. So the changes. If individual carrying the gene A are more successful in reproduction than the individual carrying the allele A, the frequency attempt to be greater than that of A. If the allele frequency A is the favorable allele, we try to be select the favorable alleles A containing individual than the recessive allele, so that the frequency of A becomes greater than the frequency of the recessive A in the next generations after the selections. So if plants with dominant homozygous or recessive homozygous genotypes were selected without error, the frequency of allele in selected population would be 1 or 0 respectively. Therefore, only A or A allele will be present in the next generation, that is selected allele fixed, indicating selection against remaining genotype is complete. यदि हामी चाहिँ हामीले dominant allele AA homozygous line or recessive homozygous line मात्रै हामी सिलेक्सन गर्यौं भने के हुन्छ भन्दा यदि हामीले चाहिँ dominant homozygous सिलेक्सन गर्यौं भने the allele frequency becomes 1 हुन्छ यदि चाहिँ homologous recessive allele selection गर्यो भने the allele frequency for the dominant A becomes 0 हुन्छ so यसले चाहिँ के जनाउँछ भन्दा next generation मा either A allele or either recessive A allele will be present not the both so that's this indicates the selection against remaining genotypes is complex if in such cases disadvantage in reproduction that is selection differential is equal to 1 and fitness or adaptive value becomes 0 for genotype that are not selected यदि यस्तो भयो भने के हुन्छ हामीले चाहिँ homozygous A selection गर्यो dominant भने के हुन्छ भने जुन not selected जुन छ हैन भनेको द लेफ्ट ओभर भनेको रेसेसिभ एलिल को चाहिँ एडाप्टिभ भ्यालु चाहिँ के हुन्छ अहिले पनि 0 हुन्छ किनकि दैट इज नट बीन सिलेक्टेड दैट विल नट बी एडाप्टेड तर homozygous को एडाप्टिभ भ्यालु चाहिँ 1 हुन्छ and the selection differential becomes 1 yes becomes 1 र adaptive value becomes 0 for the not selected completely unselected so generally yes is less than 1 always selection difference is not becomes 1 it is not possible to identify genotypes with certainty due to dominance and less than 100% heritability and consequently selection is expected to change gene frequency rather than to eliminate one or other alleles so के हुन्छ भन्दाखेरि यो चाहिँ दिस इज अफ आइडियल केसेस यदि हामी चाहिँ जीनोटाइप बेस्ड और हामी सिलेक्सन गर्यौं भने दैट मे ह्यापन्स तर के हुन्छ भने हामी फेनोटाइप बेस्ड सिलेक्सन गर्दै जाँदाखेरि वी क्यान नट कम्प्लिटली इलिमिनेट द alleles which is unfavorable even though it is unfavorable alleles that cannot be eliminated from the selections but हामी इलिमिनेट गर्न सक्दैनौं किनकि के भन्दा एक त देयर इज with the dominance relationships we cannot be identified with certainty that it is dominant हो या हैन भने हामी आइडेन्टिफाई गर्न सक्छौं certainly भने अर्को चाहिँ के हुन्छ भन्दा heritability is never 100% for the particular traits heritability 100% हुँदैन यसले गर्दाखेरि के हुन्छ भन्दा सिलेक्सनले चाहिँ इलिमिनेट गर्न सक्दैन means the adaptive yes never becomes 1 for the particular alleles which is being left over भने अर्को चाहिँ के हुन्छ भन्दा we can only change the frequency of the alleles dominant to recessive allele frequency that can be changed means dominant or favorable allele frequency can be increased and unfavorable allele can be decreased generation after generation of selections when S is less than 1 the rate of change in gene frequency delta Q would depend on intensity of selection and gene frequency अब yes the selection differential less than 1 भएपछि किनकि त always less than 1 हुन्छ not all completely 100% the 1 हुँदैन selection differential it is less than 1 so the rate of change of the gene frequency and जसले गर्दा के हुन्छ भन्दा gene frequency चाहिँ changes हुन्छ eliminate हुँदैन तर gene frequency for particular allele will be changes हुन्छ भनेपछि that is the delta Q for the recessive allele को लागि यदि gene frequency delta Q हो भने that will be depend on the intensity of selection or gene frequency ma bar parts they have intensity of selection yes and gene frequency Q that will determine the changes in the gene frequency delta Q when an allele is rare in population delta Q due to selection for this allele is small यदि very rare छ allele भने यदि frequently available allele हो भने delta Q becomes larger हुन्छ it is very rare so it cannot be find out easily rare means it cannot be found out easily and so the delta Q becomes very smaller as the value of Q increases due to selections the delta Q also increases reaching a maximum at about Q is equal to 0.3 in case of the dominant allele A Q is equal to 0.5 when there is no dominance and Q is equal to 0.7 in case of recessive allele A after this delta Q again declines as the favored allele becomes more frequent in populations अब के हुन्छ भने जस्तो हामी सिलेक्सन गर्दै जाँदाखेरि हामीले फेभर्ड एलिलहरुलाई सिलेक्सन गर्ने गर्छौं रेयर एलिलहरुलाई चाहिँ हामी very difficult to be changes in the frequency allele frequency change गर्न very difficult हुन्छ किनकि they are very rare and they cannot be easily be identifiable हुनस अब हामीले चाहिँ यदि हामीले frequency of the Q increase भयो भने के हुन्छ because of the selection frequency of the Q increase भयो भने delta Q also increase increases, changes in the gene frequency becomes increases. जब Q increase हुँदै जाँदाखेरि rare छ भन्दा very small हुन्छ, तर Q allele rare alleles frequency Q if increase भयो due to selection ले भने delta Q also increase हुन्छ, and it becomes maximum at about Q is equal to 0.3. कुन बेलामा डेल्टा क्यू मैक्सिमम हुन्छ भन्दा Q is equal to 0.3 भयो भने, in case of dominant allele A भयो भने Q is equal to 0.5 when there is no dominancy भयो भने, or Q is equal to 0.7 in case of the recessive allele A. So after this reaching maximum मा पुगिसकेपछि के हुन्छ भन्दा favorable allele increment हुन्छ, favorable allele becomes more हुन्छ, जसले गर्दाखेरि now again delta Q try to be declines, delta Q is now declines after it reaches to the maximum because the favorable frequencies are more there. So selection in random matching population is highly effective in increasing or decreasing the frequency of allele, but is unable to either fix or eliminate them. However, in combination with the system of inbreeding, selection is highly efficient in fixation and elimination of alleles. अब हामीले सेलेक्सनबाट चाहिँ एलिमेन्ट्सहरु एलेलहरुलाई चाहिँ एलिमिनेट गर्न दिन सक्दैनौं, फिक्सेसन पनि गर्न भेरी डिफिकल्ट हुन्छ। तर यसले चाहिँ के भन्दाखेरि सिलेक्सनले जुन एलेलिक फ्रिक्वेन्सी हो that will be changes, either one allele becomes increase in its frequency or other allele becomes decrease in its frequency, that will be happens. तर selections in combination with the inbreeding helps in fixation and elimination of the alleles. So when selection favor heterozygote, यो चाहिँ homozygous case भयो भने, when the selection favor heterozygotes, both the allele are retained in the populations. Homozygous भयो भने त के हुन्छ भन्दा either one allele will be present there, another will be reduced there, हैन। so complete ideal भयो भने त completely eliminate पनि हुन्छ। तर यदि हामीले selection चाहिँ heterozygous मा गर्यौं भने चाहिँ के हुन्छ भन्दा in the next generation also both allele becomes retained. So Q and P reach equilibrium when Q is equal to SA upon SA plus S dominant A, where S recessive A and S dominant A are the selection intensities against homozygous and homozygous recessive and homozygous dominant genotypes. Selection intensity हो यो। So P र Q चाहिँ equilibrium कतिखेर पुग्छ भन्दा when Q is equal to selection intensity for the recessive allele A upon selection intensity of recessive plus selection intensity for the dominant allele. It becomes equilibrium. The value of Q is often differed from 0.5, although at Q is equal to 0.5, the frequency of heterozygous, the most favored genotypes, is the maximum. Selection will be unable to fix or eliminate allele in case of character governed by the several genes also. Not by a single genes governed. Several gene governed गर्ने characterहरुमा पनि selection cannot fixed or eliminate the alleles. So for a genes, delta Q is equal to greatest when Q is equal to 0.5, since gene action assumed to be additive in the polygenic cases. Several gene polygenic cases भयो, polygenic cases मा हामीले के assume गरे हुन्छ भन्दा every genes have a small and additive effect. So there is the additive gene action being exist भयो भने के हुन्छ, for a gene delta Q is equal to greatest when Q is equal to 0.5, just like as the maximum when there is no dominancy. With Q less than 0.5 or Q greater than 0.5, the delta Q for a gene would becomes smaller. अब चाहिँ Q को value 0.5 भन्दा बढी या 0.5 भन्दा कम भयो भने के हुन्छ, delta Q for a gene becomes smaller, and delta Q would become smaller as the number of gene governing the character increase. किन भन्दा जति यसको चाहिँ maximum Q is equal to 0.5 भन्दा तल या माथि हुन्छ, the number of genes governing their character will be increase हुन्छ. So delta Q also become smaller at that times. With the increase in frequency of desirable allele, the frequency of desirable genotype will also increase automatically. New genotypes will also appear producing more extreme phenotypes, so that mean of population will change in the direction of the selections. Polygenic character हामी सिलेक्सन गर्यौं भने mean frequency कतातिर मुभ गर्छ भन्दा mean frequency of move population को mean र selected population को mean changes आउँछ, so that the movement of the mean value for the phenotype for that particular trait will be moved toward the direction of selection. जुन direction मा selection गरिन्छ त्यो direction मा चाहिँ mean value move भनिएको छ. So in case of the quantitative character governed by only few genes, with a large effect, that is the oligogenic effect, delta Q would relatively large and desirable allele may become fixed after some generation of selections. अब चाहिँ oligogenic or qualitative characters a few genes having a large effect government character हुन्छ त्यसमा delta Q becomes larger and it will be fixed after some generation of selections. That will happens. The progress under selection for quantitative character is retarded by selection there must be have some genetic progress genetic advancement, but it will be retarded by because non-additive gene action not only that the additive gene action being happens, non-additive gene action also being happen for the acceleration of that particular traits. Or every characters they do not have a high heritability or 100% of variability. There is a low heritability that is less than 100% for the quantitative characters, and the limit on permissible selection intensity. Permissible selection intensity is very limited, and dependent upon fecundity, that is reproductive capacity of the species. The selection intensity is dependent on the reproductive capacity of that species. एकदम फेकुन्डेन्ट छ भने त सिलेक्सन इन्टेन्सिटी चाहिँ लो हुन्छ, एकदम लो फेकुन्डिटी छ भने सिलेक्सन इन्टेन्सिटी फेरि हाई हुन्छ. हामीले एकदम पर्टिकुलरली चाहिँ very few number of individual has to be selected or very high number of individual has to be selected. That will be dependent on the fecundity. So because of that, the progression or selection will be retarded in the quantitative traits. When two characters have to be selected simultaneously, the selection intensity for each the two characters has to be relaxed to accommodate selection for the others. अब यदि हामी चाहिँ दुईटा क्यारेक्टरलाई सँगसँगै हामी सिलेक्सन गर्नु छ भने एउटा क्यारेक्टरलाई रिल्याक्स गरेर अर्को क्यारेक्टरको सिलेक्सनलाई हामी चाहिँ एकोमोडेट गर्नुपर्ने हुन्छ. सो फर एक्जाम्पल इफ सिलेक्सन इज प्र्याक्टिस फर थ्री क्यारेक्टरलाई सिलेक्सन गर्यो भने फर द 5% पपुलेसन इज सेभ्ड इन द इच जेनेरेसन्स र हामीले इन्टेन्सिटी कति राख्यौँ भने 5% आफ्नो इन्टेन्सिटी हो र डिफरेन्सियल हो भने हैन, इफ वी प्रोभाइड दैट द सिलेक्सन इन्डिभिजुअल इज 5% is 5 out of 100 so the intensity of selection for any one character would be always under root 3 cube under root upon 0.05 or 36% of the 5%. 36% चाहिँ रिल्याक्सेसन गर्नुपर्ने हुन्छ. Selection intensity चाहिँ हाम्रो 36% हुन्छ, not 5%. So in this way we have to be relaxed on characters that have to 36%, another character may have a 5% of selection intensity to be done. The fitness or adaptive value. A fitness value is the reproductive success. So adaptive value reproductive ability of the traits of the individuals. So fitness is relative reproductive success in its intelligence. The rate of reproduction for a genotype is the average number of progeny produced by each individual of that genotypes. Rate of reproduction for a genotype average number of progeny produced by individual or that genotype. That gives those progeny which fail to reproduce are not considered while determining reproduction rates. अब यसमा चाहिँ हामी के गर्छौँ त त्यस्तो इन्डिभिजुअललाई मात्रै हामी कन्सिडर गर्छौँ those they can reproduce the individual or offspring which can survive in or which have a survivability or viabilities in the to be survived in the next generation. र those which are fail to produce those progeny which are failed to reproduce are not being considered by determining the reproduction rates. Progeny that has that must have the ability to produce the viable offspring. Viable हामी reproductive rate calculation हामी त्यसलाई फिट गर्ने गर्छौँ. The fitness or adaptive value that is W may have any value between 0 to 1 for the most productive genotype and 0 for the lethals. There is no reproductivity. W is equal to 1 if it is have a 100% or the highly productive genotypes को लागि भने W is equal to 0 for the lethals genotype को लागि. So W is equal to 1 minus S. S is the selection coefficient. Selection against a genotype may operate on gametes, the haploid, or on zygote, the diploid, or both, depending at which stage gene expression affect survivability. So our selection हामी gamete तो haploid stage में कर सकते हो, selection हामी diploid stage में, that is zygote stage में कर सकते हैं, or in both stage, that is depending on at which the gene एक्सप्रेसन एफेक्ट द सर्भाइभल्स। त्यो जिन एक्सप्रेसन ह्याप्लोइडमा चाहिँ एफेक्ट गर्छ भने ह्याप्लोइडमा सिलेक्सन गर्छौँ, डिप्लोइडमा एफेक्ट गर्छ भने डिप्लोइडमा गर्छौँ, बोथमा एफेक्ट गर्छ भने दुवै स्टेजमा हामी चाहिँ सिलेक्सन गर्न सक्छौँ। So in this way the selection can may be practiced at the gametic level or the zygotic level as gametic selection and zygotic selection selection practice गर्न सक्छौँ। The gametic selections, when selection acts on gametes or on haploid phase of the life cycles, just like is the fungi, Neurospora that having of haploid stage. त्यसमा चाहिँ हामी चाहिँ when selection act on the gametes, in most of the fungi the main part of the life cycle is haploid, so selection is the gametic. जसको चाहिँ म्याक्सिमम साइकलहरु लाइफ साइकलको चाहिँ म्याक्सिमम स्टेज चाहिँ ह्याप्लोइडमा एन्डिङ हुन्छ, त्यसमा हामी चाहिँ ग्यामेटिक सिलेक्सन प्र्याक्टिस गर्छौँ जस्तो फंगाइहरुमा। अनि अर्को हुन्छ जाइगोटिक सिलेक्सन। जाइगोटिक सिलेक्सन चाहिँ हामी हायर अर्गानिजमहरुमा जाइगोटिक सिलेक्सन गर्छौँ। दोज दे स्पेन्ड देयर लाइफ साइकल्स इन अ जाइगोटिक स्टेजमा। मोस्ट अफ द life cycle is spent in the diploid condition and they also have a certain short haploid life cycles also. Zygotic selection हुँदाखेरि मल्टी अर्गानिजम्सहरु अथवा हायर अर्गानिजमको चाहिँ सर्ट gametic cycle पनि हुन्छ अथवा haploid cycle पनि हुन्छ, but most of the cycles is the life cycle is spent with the diploid अथवा gametic stage. So most higher organisms are diploid and their haploid phase is only short-lived and totally depend on the diploid phase. So selection takes primary in the diploid or zygotic stage. There are three possible genotypes for a single gene differences. If a single gene locus and having two alleles, three genotypes may be produced from there. So dominant A, heterozygous A, and recessive A. So that the effectiveness of selection depend, among other things, upon the degree of dominance. If complete dominance exists and selection occurs only against the recessive allele A, the heterozygote is shielded from selection and harmful or even lethal recessive genes may persist for many generations. यदि हामीले चाहिँ डोमिनेन्स कम्प्लिट डोमिनेन्स एक्जिस्ट भयो भने के हुन्छ भन्दा हाम्रो सिलेक्सन चाहिँ होमोजाइगसमा पनि हुन्छ, हेटेरोजाइगसमा पनि सिलेक्सन हुने भयो, किनकि देयर इज अ कम्प्लिट डोमिनेन्स बाइ दिस डोमिनेन्ट एलेल, सो वी विल सिलेक्टेड बोथ अगेन्स्ट दिस रिसेसिभ एलेल्स को हामी यसमा अगेन्स्ट हामी यो दुईटा हामी सिलेक्सन गर्छौँ। यसलाई चाहिँ सिलेक्सन गर्दाखेरि के हुन्छ भन्दाखेरि जुन यो रिसेसिभ एलेल छ, रिसेसिभ एलेल ए, that also being exist, the recessive lethal allele that being exist for many generations. Yeah, that comes to genes. When dominance of advantageous allele is incomplete, यदि चाहिँ dominance incomplete dominance छ भने heterozygous will show the effect for a deleterious genes since the heterozygous phenotype is at least partially deleterious. So heterozygous के देखाउँछ भन्दा deleterious genes expression show गर्छ. If dominance is absent completely and heterozygote has a phenotype exactly intermediate to that of the two homozygotes, its selection coefficient will be exactly half that of the deleterious homozygotes. यदि चाहिँ completely absent भयो dominance भने के हुन्छ भन्दा heterozygosले कस्तो शो गर्छ भन्दा in between of this dominant homozygous and recessive homozygous को in between expression गर्छ heterozygous. So intermediate phenotypes. So its selection coefficient will exactly have that of deleterious homozygotes. So heterozygous को चाहिँ selection के हुन्छ भन्दा 1/2 of dominant and recessive homozygous. So diagnostic selection may be again the recessive phenotypes, that is against AA गर्यो भने it will fix the dominant allele AA. Against dominant phenotype गर्यो भने के हुन्छ, dominant phenotype हामी चाहिँ against selection गर्यो भने के हुन्छ भन्दाखेरि the recessive allele being fixed and the again the selection being against dominant homozygous हुन्छ भने in favor of heterozygous गर्यो भने against recessive allele and dominant homozygous and it will fix the heterozygous genotype will fix in the next generation when we practice the zygotic selections. So in this way the selection can be practiced at the haploid level stage or the zygotic or diploid level stage मा हामी सिलेक्सन गर्न सक्छौँ। अर्को फ्याक्टर भनेको इनब्रिडिङ हो। You are well known about this inbreeding things, you know. So mating between individuals sharing a common parent in their ancestry. Inbreeding means यदि हामी चाहिँ consanguineous mating भयो भने that will show the inbreeding. त्यो भनेको चाहिँ common parent in their ancestral stream as well. त्यस्तो इन्डिभिजुअल बिचमा इन्भेटिङ गर्यो भने that will show the inbreeding date. And in a small population, a certain amount of inbreeding is bound to occurs. So reduces in the proportion of that. It is a population सानो छ भने के हुन्छ भने there is a higher chances of the consanguineous mating. So the inbreeding will be bounded there. Say. Reduces the proportion of the heterozygotes or heterozygosity, and if the proportion of homozygous or homozygosity will be increased. In the monoecious or hermaphrodite species, the rate of decrease in heterozygosity per generation becomes 1 by 2n, half of the population. हुने गर्दछ 1 by 2n decrease in the heterozygosity. In the same way, the increase in the homozygosity. In the dioecious species or monoecious species, where self-pollination is prevented, that is in the cross-pollinated or dioecious species or monoecious with of cross-pollination. Rate of decrease in heterozygosity per generation is equal to 1 upon 2n plus 1. Thus, in a small population, even with the strict random mating, the frequency of homozygotes increases while that of heterozygote decreases due to inbreeding. If the population is smaller, the inbreeding will have a greater effect on the changes in the homozygosity and heterozygosity. Because there is a strict, even though we practice a random mating structure, there also exists some consanguineous mating. Individuals, they may have any kinds of the relationship in their ancestral history. They may have a parental sharing. So inbreeding would be happens there, even though we make a random mating in the small populations. Okay. Is there anything?